Hello, Bacho. Welcome in the platform of Biology again with the NCERT explanation of chapter Excretory System of paragraph 19.3. Before 19.3, we have already gone through paragraph 19.2, 19.1, as well as the introductory para. Basically, in this para 19.3 of excretory system, we are going to deal with the functions of tubule. The functions of tubules. Yes, I'm talking about the tubules PCT, loop of Henle, which is called Henle's loop, DCT, and collecting valve. So the heading of paragraph 19.3 is the functions of tubule. You can see the NCERT interface beta. It is the interface of NCERT, paragraph 19.3. Heading is the functions of tubule, in which we are going to talk about first the function of TC, the proximal convoluted tubule. After that, the function of Henle's loop. After that, the BC's function, and lastly, the function of collecting valve. This was the main content of our today's lecture, the paragraph 19.3. So, what we are going to study in this? First, the function of PCT, which is called proximal convoluted tubule. After that, we'll talk about the function of Henle's loop, or which we can also say the loop of Henle. After that, we will talk about the DCT, which is the distal convoluted tubule. And lastly, the collecting valve. This is all on today's content. Is it clear? Now, आगे देखते हैं. Now, see, when you start talking about the first tubule of the nephron, which is the proximal convoluted tubule. This tubule is the first tubule of nephron having the property of maximum reabsorption. It means here there will be maximum reabsorption take place. And reabsorption of what? Yes, the reabsorption of basically the sodium, the potassium, water, glucose, galactose, and amino acid also. And also some amount of carbonate ion. These things will be reabsorbed by proximal convoluted tubule of a nephron. And this is the part when the maximum reabsorption will take place. It means we can also say that PCT is a major organ which is responsible for the maximum reabsorption. Now see on the diagram, this is the interface showing the maximum reabsorption going on in this PCT. Now I am drawing, this was the glomerulus, this was the glomerulus, this was the PCT. Basically PCT is a highly convoluted structure, zigzag structure, but for explanation I am drawing here simple tubule. Okay. This is basically the PCT. Now, what will going to happen in this PCT? These things will be reabsorbed from the filtrate inside the body. It means those things which are useful for the vein will be reabsorbed by PCT and given into the bloodstream, not released into the urine. And these things which are going to be reabsorbed by PCT will not take part in the formation of urine. It means here filtrate has come into the capsular space. Now this filtrate will come forward, and during the way, these things like sodium, like potassium, like water, like glucose, and many things will be reabsorbed by PCT. Now let's see the detailed mechanism, how this reabsorption of sodium, potassium, water, glucose, galactose, and many more are being reabsorbed from the PCT. What are the things which are responsible inside the PCT for the reabsorption of these following things? Now see the detailed mechanism. Okay. देखिए होगा डिटेल मैकेनिज्म कैसे reabsorption हो रहा है PCT के अंदर. What is responsible for the reabsorption in the PCT? See here. Now here I am drawing only the PCT. This is the lumen of PCT. Here was the glomerular filtration glomerulus. This was the BCP. There are some specialized simple cuboidal brush border epithelial cells on the inner wall of BCP. This is the inner wall of BCP containing some special tissues which are simple cuboidal brush bordered epithelial tissues. Epithelial tissue. And we know that where there will be the brush border epithelial tissue, there must be microvilli on it. It means this tissue having microvilli, and these microvilli are responsible for increasing the surface area. And for what? They are increasing surface area for what? For having facilitate more and more reabsorption. For having more and more reabsorption. These are the microvilli. These are the microvilli finger-like structures present on this simple cuboidal brush border epithelial tissue, and these are actually the tissues which are responsible for the reabsorption. Now see how. First, what will happen? The sodium present inside this tissue will come into the bloodstream. These are the bloodstream area. This is the lumen area of this tissue. I am writing here. These are the lumen of this tissue. This is the bloodstream area. Now what will happen? The sodium, the concentration of sodium present inside this specialized microvilli containing brush border epithelial tissue will give the sodium into the bloodstream. It means this sodium must get into the bloodstream. But how? By active transport. Sodium is being given into the bloodstream through active transport. But against the concentration gradient. It means here is the less concentration of sodium inside this tissue, and here is the more concentration. But then also these sodium are given forcefully into the bloodstream against the concentration gradient. So first sodium has came into the bloodstream from this tissue. Now some sodium concentration which is present inside the lumen of the PCT will come inside this cell. Why? Because after giving this sodium concentration to the blood, here the concentration has become very much low. So the higher concentration sodium will come inside the tissue where there is no concentration of sodium. Secondly, this sodium will come inside the cell. And along with sodium, water will also come inside the cell and after into the bloodstream. And water has this privilege that water can also come through a cell inside the bloodstream or through a tight junction. What is this tight junction, beta? This tight junction is basically the minute gap between these specialized brush border tissues. Okay, so water can come either through by the cell into the bloodstream and also can come from these tight junction. This is the tight junction, the minute gap between these epithelial tissues. Okay, after this, we know that sodium has been entered into the cell. There must be a transport carrier. There must be a transport carrier, or we can say the carrier transport. Okay, and these carrier transports facilitate and help the sodium to enter into the cell. And along with the sodium, from this carrier transport, glucose, galactose, and amino acid also. Come this same carrier transport along with sodium. So that's why glucose, galactose, and amino acids are called co-transport of sodium. It means mainly the sodium is being transported from the carrier transport, but along with that साथ साथ में वो भी आएगा. ठीक है, जैसे होता है ना कि हम कहीं जा रहे हैं, हम चलो हम जाए, चलो साथ में सोडियम तो आएगा एक्शन के अंदर, साथ में वो ट्रांसपोर्ट के रूप में ग्लूकोज, गैलेक्टोज और अमीनो एसिड भी एंटर करेंगे उस सेम को ट्रांसपोर्ट कैरियर के थ्रू. अब ग्लूकोज भी आ गया, गैलेक्टोज भी आ गया और अमीनो एसिड भी आ गया. तो अब ग्लूकोज, गैलेक्टोज और अमीनो एसिड्स आर गिवन इनटू द ब्लड स्ट्रीम through again there will be again a carrier transport. This carrier transport is called GLUT2. By the help of this blood to carry transport, the glucose, galactose, and amino acid inside the epithelial tissue will be given into the bloodstream. Okay. After that, talk about potassium. These potassium are also given into the cell and after into the bloodstream. Only 80% of the potassium is reabsorbed by the PCT and rest 20% will be reabsorbed forward in DCTs and loop of Henle वगैरह. ठीक है. 80% सिर्फ DCT में होने वाला है, बाकी 20% आगे चल के reabsorb हो जाएगा. तो अब सबका परसेंट देख लेते हैं. Sodium has been reabsorbed 60 to 70% only. टोटल सोडियम में से 60-70% ही रिएब्सॉर्ब होता है किसी भी. वाटर भी 60 से 75% के आसपास रिएब्सॉर्ब होता है. ठीक है. और ग्लूकोज, गैलेक्टोज और अमीनो एसिड ये टोटल हो जाते हैं. मतलब 100% ऑफ द ग्लूकोज, गैलेक्टोज एंड अमीनो एसिड आर reabsorbed by the proximal convoluted tubule. आया समझ में? It means I am making a flow chart बेटा here. सोडियम कितना रिएब्सॉर्ब होता है? 60 से 70%. Yes. Potassium only 80% here. Glucose 100% total. Galactose it is also 100% reabsorbed from the proximal convoluted tubule. And amino acid this is also reabsorbed completely. इसीलिए यूरिन में ये तीनों नहीं पाए जाते जनरली. लाइपोसोडिया वगैरह इसमें पाए जाते हैं लेकिन जनरली नहीं पाए जाते क्योंकि सारे के सारे किसी के द्वारा reabsorbed हो जाते हैं. This was basically the detailed mechanism that how this reabsorption is being proceeded by the proximal convoluted tubule. ठीक है. ध्यान से इसको नोट कर लीजिए. अब आगे देखते हैं. बेटा लूप ऑफ हेनले की बात करते हैं. आफ्टर TCP the second tubule which we are going to talk about is loop of Henle or Henle's loop. Basically there are two limbs of loop of Henle. The descending limb and the ascending limb of loop of Henle. It means see on the diagram here. We have talked about this glomerulus, this PCP. Now we are talking about this loop of Henle. This is better the loop of Henle. We are talking about the loop of Henle. PCP has been covered by us. Glomerulus also covered by us on the first step of urine formation, which was glomerulus filtration, which is also known as ultrafiltration. Now we talk about basically the loop of Henle. See this downward limb, which is going downward, filtrate will come downward. This limb is called the descending limb. The descending limb of loop of Henle, LOH. I'm writing it short, loop of Henle. It is the descending limb of loop of Henle. Here the filtrate will come downward because filtrate is coming in this direction. It will come downward. Now here the filtrate is starting moving upward. So this is the ascending limb of loop of Henle. This is the ascending limb. Ascending limb of loop of Henle. Okay. Now see what happened. ECG में reabsorb हो गया जो होना था हमने देखा. अब कुछ यहाँ पर भी reabsorb होगा. देखो क्या. Descending limb is permeable for water. This is permeable for water, but impermeable for solutes like sodium, chloride, this stuff. Okay. This is impermeable for solute. It means यहाँ पर वाटर तो रिएब्सॉर्ब हो जाएगा क्योंकि वॉल से कंटिन्यूटी है लेकिन सॉल्यूट नहीं होगा. इट मींस जैसे जैसे फिल्ट्रेट डाउनवर्ड जाएगा डीप इनटू द लूप ऑफ Henle, वाटर रिएब्सॉर्ब होता चला जाएगा और ये फिल्ट्रेट कैसा होता चला जाएगा? कंसंट्रेटेड. किसी भी चीज से सॉल्यूट रहने दो, पानी निकालते जाओ तो गाढ़ा होता जाएगा ना कंसंट्रेटेड. इट मींस जब फिल्ट्रेट इज कमिंग डाउनवर्ड द फिल्ट्रेट will become concentrated. और ठीक उल्टा भी बनेगा रहता है. ascending limb ये वाटर को नहीं परमिशन देता है एब्जॉर्ब करने. इट इज impermeable for water. I am talking about descending limb. इसीलिए the ascending limb and the descending limb both are opposite in the manner of reabsorption. दोनों को उल्टा है reabsorption. ये वाटर को करेगा, ये नहीं करेगा. ये बताओ फिल्ट्रेट अब धीरे धीरे ऊपर जाने वाला है. इट इज गोइंग अपवर्ड इनटू द ascending limb of loop of Henle. यहाँ water is not being permeable, but solute are permeable. यहाँ solute permeable हो गया. So the filtrate is becoming dilute and dilute and dilute. जैसे जैसे ऊपर जाएगा dilute होता जाएगा, क्योंकि पानी से तो absorb होगा नहीं होगा, solute होगा, तो फीका पड़ने लगेगा. तो here in ascending rate of group of filtrate, the filtrate will become dilute. Okay, so ये था एक option is loop of filtrate. अब this filtrate is moving upward into the DCT. बट ऊपर डीसीटी में ले जाने से पहले मैं एक और चीज बता देना चाहता हूँ. इफ आई एम राइटिंग हियर, मान लेते हैं आप फिल्ट्रेट था, उस टाइम फिल्ट्रेट को मैंने ए कहा, ये फिल्ट्रेट यहाँ पहुंच गया बी, बी यहाँ कह दिया, यहाँ पहुंचा सी, यहाँ फिर पहुंचा डी, तब तो डीसीटी मैटर करेगा. तो थोड़ा बात करते हैं नेचर ऑफ द फिल्ट्रेट एट दिस फोर different positions. Now we are talking about the nature of the filtrates in this four different positions. वैसे तो nature of filtrate हमको पता था यहाँ, very much similar to the composition of plasma. लेकिन हम देखते हैं ये PCT की starting हो रही, अभी PCT में enter करने वाला है. तो PCT में enter करते हैं, इसमें से water solids दोनों ही के दोनों reabsorb होने वाले हैं. इसका मतलब इसमें कोई अंतर नहीं है. ऐसा तो है नहीं water comes from there. So this both will be isotonic. Both will be isotonic. This three and this. Now, यहाँ पहुँचा, तो जैसे इसमें आया लूप ऑफ फैले के डिसेंडिंग लूप फिलिंग में, वैसे यहाँ पे वाटर भी एब्सॉर्ब होने लगा और सॉलिड नहीं हो. इसका मतलब इससे ये ज्यादा गाढ़ा हो जाएगा, इसका मतलब ये हाइपरटोनिक हो गया. The filtrate C is hypertonic in compared to A and B. And A and B are both isotonic, same osmolarity होने वाले. अब हाइपरटोनिक हो गया. अब ये आगे बढ़ा तो ascending लूप में क्या हुआ कि वाटर इज नॉट परमेएबल, वाटर नहीं रिएब्सॉर्ब हो सॉलिड में, तो ये फीका पड़ने लगा ना, ये हाइपोटोनिक हो जाएगा. हाइपोटोनिक इन कंपेयर टू C. बट B और C लगभग एक osmolarity हो जाने वाले, सेम ऑस्मोलैरिटी के होने वाले हैं. इसका मतलब ये दोनों भी आपस में आइसोटोनिक हो जाएंगे. They have the same osmolarity. The nature of this filtrate at this four different positions, I think absolutely clear. Now it is entering into the DCT. अब देखिए बेटा DCT. Now we are talking about basically DCT, the distal converted tubule. It means the filtrate has already reached past the limbs of loop of filtrate and been entered into the DCT. Filtrate यहाँ पहुँच चुका है DCT के पास. Now, when we talk about the DCT beta, DCT basically shows the conditional reabsorption of sodium. ये सोडियम का conditional reabsorption कराने वाला है. ये समझेंगे बेटा, what is the meaning of this conditional reabsorption of sodium by the DCT? ये secrete कराएगा क्या? Potassium. It will also secrete potassium and also secrete the H plus ion and ammonia, and by secreting this H plus ion and ammonia, it will maintain the pH of body fluid. These are the basic functions of DCT. लेकिन इन सारे functions हम समझते हैं कि DCT is how actually doing this all these functions and maintaining the pH by releasing, secreting H plus ion and ammonia. Now see in detail. Now see better. We have seen that DCT has basically two functions: the conditional reabsorption of sodium and secreting potassium ion, and also secretes H plus ion and ammonia to maintain the pH of the body fluid. Now see how. Basically, the hormone aldosterone, which was released by the adrenal cortex, the cortex part of adrenal gland. This hormone is responsible for both these functions of DCT. The conditional reabsorption is also being done by the aldosterone hormone, which was being secreted by the adrenal cortex, and secondly, the secretion of potassium in the body is also being helped by the aldosterone hormone, which was released by the adrenal cortex. Now see how. I am telling this hormone aldosterone, which was basically released by the adrenal cortex. Okay. This was released by adrenal cortex. I am telling that this hormone will facilitate both of these functions, which are the reabsorption of sodium, and secondly, the secretion of potassium. Let's see how. Let's start with the reabsorption of sodium. Basically, what happens? Basically, the reabsorption of sodium beta is being preceded in the presence of angiotensinogen. Basically, angiotensinogen is a protein. This angiotensinogen is a protein which is being converted into angiotensin 1 in the presence of renin. In the presence of renin, this angiotensin is being converted into angiotensin 1, and further this angiotensin 1 is again converted into angiotensin 2 in the presence of ACE, which is basically the angiotensinogen converting enzyme, which is mainly released in large amount by lungs. Okay. Now see that after converting into angiotensin 2 by the angiotensinogen converting enzyme, which is basically released in a large portion from the lungs, this angiotensin 2, we know that this angiotensin 2 is a huge and a very powerful vasoconstrictor. So this angiotensin 2 will stimulate the adrenal cortex, basically the cortex part of adrenal gland, and then this adrenal cortex will release the aldosterone, and this aldosterone will lead to the reabsorption of sodium inside the DCT, because this aldosterone, which was released by the adrenal cortex after the stimulation of angiotensin 2, will act on the DCT and facilitate the reabsorption of sodium. So this was basically how aldosterone is playing the role inside the DCT for the conditional reabsorption of sodium. Now see what about the secretion of potassium? Basically, this potassium is being filtered in the glomerulus, but after moving forward, this potassium is reabsorbed by PCT. We have seen in the PCT that PCT was reabsorbing 90% of the potassium. After this, if there will be an increase in the concentration of potassium in the body, there will be increase in the concentration of potassium in the body. The body will release. The body stimulates the adrenal cortex, and adrenal cortex releases the aldosterone, and this aldosterone acts on both DCT as well as the collecting duct, and both this DCT and collecting duct will start secretion of potassium. This is how the aldosterone is again helping in the secretion of potassium. Now see that the conditional reabsorption of sodium and the secretion of potassium was being done by this mechanism by the help of aldosterone. Now see how the secretion of H plus ion and ammonia is going to maintain the pH of the body fluid. Now see that how the secretion of H plus ion and ammonia will maintain the pH of the body fluid. Now see, if we assume that this is the body and this is the collecting duct, this is the collecting duct where there will be the ingredient. So the H plus from this body, the bloodstream, has been given into the collecting duct. It means we have already maintained the pH of the body. But what about this urine? Here is a urine in collecting duct. We have गलती से बेटा हमने बॉडी के H प्लस को हटा करके यूरिन के अंदर ये इसको मेंटेन कर लिया बॉडी, लेकिन गलती से हमने ये नहीं ध्यान दिया कि यूरिन में भेजने की वजह से यूरिन की पीएच बढ़ गई, एसिडिक नेचर बढ़ गई, तो ये दिक्कत कर सकती है हमें. ठीक है, सो वी हैव ऑलरेडी मेंटेन्ड द pH of the body, but we have गलती से बेटा increase the acidic nature of the urine. So now, what about urine? How we have to maintain the urine body fluid? How to maintain the pH of urine? So body will again secrete ammonia in this collecting up to the urine. Now this ammonia will convert into NH4 plus, which is basically the weak acid. It means we have weakened the acidic nature of urine also. We have successfully weakened the acidity of the urine. जो कि गलती से हमने बढ़ा दिया था उसको NH3 को सिक्रीट करा करके NH4 which is basically the weak acid हमने यूरिन की कंसंट्रेशन बेसिकली pH को मेंटेन कर ली. अब किसी तरह की दिक्कत नहीं होने वाली. अब बेटा ये इंटरफेस देखिए पूरा का पूरा 19.3 पीसीबी का फंक्शन बीसीबी का फंक्शन कनेक्टिंग का फंक्शन लूप ऑफ एलए का फंक्शन टोटल हमने कवर कर लिया. ओके, तो आई होप दिस concepts are completely completed.